Let's pick up with activity 3 on page 84. In order for us to simulate the transdermal patch, we're going to use a strip of dialysis tubing. Now, it may not look like it, but this, while it feels like and looks like a piece of cellophane, actually is a tube. It's open at this end and it's also open at the other end. And in order for us to actually open this up and fill this tube, we will need to get it wet. Once we do that, we're going to take some string and we're going to tie off one end, and then we're going to fill up our dialysis tubing with some fluid that will be similar to cytosol or the cytoplasm on the inside of the cell, and then we're going to tie off the other end of that. So for ease of presentation, I went ahead and I made one of those simulated cells. So this is what it looks like. You can see that it's got some string on the end and the cell itself is all filled up. So for us to be able to take a look at the medications, I'm going to just set this on a piece of plastic wrap. We now need to set up our patches, and so for our patches we're going to use a little bit of filter paper, and so there is one patch, and we are going to set up another patch, and we're going to make observations about two different medications. The first medication that we'll use is blue, and this is medication A, and we're going to apply this to the patch. Remember that as we apply this in any patch here or in one transdermal patch, we are creating an area of high concentration gradient inside of that patch, while on the inside of the cell we know that there's a low concentration gradient. We're going to do the same thing with our second medication, which is medication B, and we're going to put that on our second patch. One of the questions that we want to answer is our membrane permeable to medication A, and is our membrane permeable to medication B? So we are going to let this sit for a few moments, and then we'll be back to see what happens.